In this video, we will be discussing about horizontal gene transfer, a fascinating mechanism by which genetic material moves between organisms in ways other than traditional inheritance. We define horizontal gene transfer as the non-reproductive movement of genetic material between organisms, occurring independently of parent to offspring inheritance, which is vertical transmission. So before we dive into horizontal gene transfer, we should first understand what vertical gene transfer is. We see vertical gene transfer as the transmission of genetic information from parent to offspring. Here in this animation, we can see how it happens. We see from parent to offspring there is a transfer of DNA. Within these diagrams, the yellow one depicts the DNA. And this happens in most organisms, whether through sexual reproduction or asexual reproduction, where DNA is passed down along the generational line. In contrast, horizontal gene transfer breaks this rule. Here DNA moves laterally. We see in the animation the genes or DNA gets transferred between the organisms of the same generation, and sometimes even across different species as shown in the animation. A transfer between bacterium A to B or B to C or A to D. So different species are coexisting at the same time during the transfer. This process is especially common in bacteria, and it has profound implications for evolution and medicine. Now let's get into the details and see the whole horizontal gene transfer process. We see the horizontal gene transfer is driven through three main mechanisms: transformation, transduction, and conjugation. First of all, we will begin with the transformation. This is the process where a bacterium takes up the free environmental DNA from its surroundings. But for this to happen, the bacterium must first develop competence. Basically, the ability to bind and import DNA. That is the competence. Competence can be of two types: natural or artificial. In natural competence, certain bacteria like Bacillus Streptococcus or Neisseria are genetically programmed to do this. They use specialized DNA binding proteins and uptake machinery. Once the double-stranded DNA is taken up, usually only a single strand actually enters the cytoplasm, while the other strand is degraded. On the other hand, artificial competence is sometimes scientists induce in the lab. For example, by treating cells with calcium chloride, applying a heat shock, or using electroporation. This allows double-stranded DNA fragments to cross the cell membrane and enter the cytoplasm. Once the DNA fragment is inside, it doesn't just float around. It has to integrate into the bacterial chromosome. This happens by homologous recombination, usually mediated by proteins like archaea. And when this recombination is successful, the bacterium becomes a stable recombinant, carrying and expressing the new genetic material. Now moving on to the second mechanism of horizontal gene transfer, that's transduction. This is phage-mediated DNA transfer, meaning viruses that infect bacteria called bacteriophages are the vehicles carrying host DNA from one cell to another. There are two main types of transduction: generalized and specialized. First, let's see the generalized transduction. In this mechanism, an error occurs during phage assembly. Instead of packaging only viral DNA, the phage accidentally packs a random fragment of host bacterial DNA into its capsid. When this defective phage infects a new host, it injects that bacterial DNA rather than the viral genes. This DNA can then undergo homologous recombination with the recipient's chromosome, leading to a stable recombinant bacterium. Then we have the specialized transduction. This happens because of an excision error during the lysogenic cycle. Normally, we see a prophage, that is the viral DNA integrated in the bacterial chromosome, excises cleanly when switching to the lytic cycle. But sometimes the excision is sloppy, and the phage DNA carries along adjacent bacterial genes. So the new phage particles now contain the mix of phage and host DNA. When these phages infect another bacterium, the carried bacterium genes are inserted, usually via integration, at the phage attachment site, ATTB. So we see basically in transduction, mistakes in the phage life cycle are what actually create opportunities for bacteria to exchange genetic material. The third major pathway of horizontal gene transfer is conjugation. This is plasmid-mediated cell-to-cell contact process. Unlike transformation or transduction, here the DNA is transferred directly from one living bacterium to another through a physical connection. The classic example is the F plasmid system. In this setup, an F plus donor bacterium, which carries the fertility plasmid, comes in contact with F negative recipient. The donor extends a structure called sex pilus, which attaches to the recipient and then retracts, bringing the two cells together to form a mating bridge. Once the bridge is formed, the plasmid undergoes rolling circle replication, and a copy of plasmid DNA is transferred into the recipient. At the end, recipient becomes F plus as well, now capable of acting as a donor in future conjugation events. But conjugation isn't limited to just plasmid transfer. Sometimes F plasmid can integrate into bacterial chromosome. When this happens, the donor is called HFR, high frequency recombinant. In this case, during conjugation, not just plasmid DNA but also chromosomal genes can be transported into the recipient. These chromosomal segments may recombine with the recipient's DNA via homologous recombination. Interestingly, even though genes are transferred, the recipient usually remains F negative, because the whole plasmid is rarely transferred completely. And finally, conjugation can also involve the transfer of other elements like R plasmids, which carry genes for antibiotic resistance, making this process extremely important in clinical microbiology. So to summarize the things, we see transformation brings in free DNA from the environment, transduction transfers DNA via phages, and conjugation moves DNA directly through cell-to-cell contact. All these ultimately can lead to stable recombinant bacterium, one that carries new genetic traits. So this is what horizontal gene transfer is and its types. I hope you like the video. If you like it, give it a thumbs up. Do consider support my work on Patreon or YouTube and make sure to subscribe to the channel. Thanks.